Hello students, and welcome to my video on cell structure and function topics 2.4 through 2.7. This video is going to go over the cell membrane, why it's selectively permeable, how it maintains homeostasis, and active and passive transport. I encourage you to check out the Bozeman Science videos on this and the Amoeba Sisters. They do a really wonderful job with all topics, and also especially with this topic. Like all of my videos, this is not for profit. No money can be made off of this. This is just meant to help students who find it on the interwebs. Okay, so I'm going to look at the college guide like I asked you to do in the other videos and see what I need to know. Right? I need to be able to talk about the roles of the components of the cell membrane and maintaining the internal environment of the cell. Just like in the previous video, the structure of an organelle leads to its function. The structure of the cell membrane will lead to its function of maintaining the internal environment of the cell. And so we'll learn about that. We'll describe the fluid mosaic model of the cell as well. So let's dive right in right here with 2.4. So the cell membrane is going to be made up of phospholipids. A phospholipid has a polar head and then these non-polar tails. This would be a saturated fatty acid, and this would be an unsaturated fatty acid. Okay? They're going to line up to make the cell membrane with the polar tails facing each other and the the non-polar tails facing each other and the polar heads facing the wet environment within the cell and the wet environment outside the cell. That requires no energy, right? It's the way that they will naturally form. So let's do it one more time. We're looking at this is polar. This part goes well with water. It's hydrophilic. This part is non-polar and is hydrophobic. It is water-fearing. So here is the membrane. You can see those non-polar hydrophobic tails on the inside. They would form naturally that way. The polar heads on the outside. Embedded within that membrane it's more than just phospholipids. Right? There's going to be protein channels that allow items in and out. There's going to be markers. So this would be a glycoprotein. It's got a carbohydrate attached to a protein. And it says, hey, this is like an ID badge. This is, I work at TASM. Or you can have the same thing with glycolipids, which would be a lipid with a carbohydrate attached. You can have proteins that don't span the whole channel that are just on one side, and it might work in signal transduction pathways as well. All right, it is fluid mosaic because it's not just still like this picture shows, but in this animated GIF right here, it's moving, it's dynamic, which allows cells to change shape. And so all cells have a phospholipid bilayer membrane. All cells, plant cells, bacteria as well. Sometimes on the outside of this, there will be a cell wall, not for animal cells though. Embedded within this is going to be molecules of cholesterol that will help with the fluidity of the cell. We'll get into that later on, as well as the carbohydrates and the markers I told you about. Let's see. So we mentioned earlier the saturated and unsaturated fats. This would be a saturated fat, and unsaturated fat has a kink in it or a bend. The more unsaturated fats present, the more fluid the cell membrane would be, and the more saturated, the less fluid it would be. And so cells evolve to have the correct ratio of saturated and unsaturated fats to fit their environment. All right, cholesterol maintains fluidity at very low temperatures. It helps to keep it fluid. It helps to prevent them from packing together too tightly. And at very high temperatures, it prevents them from being too fluid to falling apart, to melting apart by interfering with phospholipid mobility. So it's kind of like a buffer. You can think about it like a buffer from Unit 1. These carbohydrates serve as ID badges, and proteins can serve as channels, receptors, enzymes, and more. Here's a good picture from Khan Academy about a cell membrane with cholesterol and without. So let's see. Oh, if it's cold, it'd be too rigid. It may break. If it's hot, it would fall apart. That's without cholesterol. All right, and then I have a picture with cholesterol coming up in just a second. Here's an example of a channel protein. This is interesting. The R groups, remember from Unit 1, that are out here would have to be nonpolar, right? Because they're dealing with the nonpolar interior of the cell membrane. So this would be all R groups that are nonpolar. Whereas the inside can be polar because that might be the molecule that's coming through. So a good example of how the structure of a molecule leads to this function. And that works with cystic fibrosis, right? This channel protein is not working for these chlorine ions, and they stay in the cell. There's relatively more chlorine ions here. That's going to actually dehydrate the area outside the cell and make this sticky mucus that can be infected for those who suffer from cystic fibrosis. Here is the image of cholesterol. So the cholesterol is going to grab onto the polar head, part of it will, and it will help to stiffen this region, and this region can be more fluid, right? And it can be in a ratio that is appropriate for the cell. All right, quiz yourself for membranes. Can you describe the fluid mosaic model of the membrane? What are the roles of cholesterol, proteins, and carbohydrates? What happens if mutations occur in those proteins? There's a really fun lab called the Cell Bubble Lab that you can just find by Googling, and it deals with like corn syrup and dish soap, etc. And it's a great way to model the fluid mosaic model. I do it with my students. It's a lot of fun. So check that out. All right, topic 2.5. Now that we've identified the cell membrane being made of these phospholipids, let's talk about the permeability of the cell membrane. And so the cell membrane is selectively permeable. It allows certain items to go right through, things that are small and nonpolar, and other items that are large or polar must use protein channels in order to pass through. Water, which is a special exception, can pass through the membrane in small amounts, but it passes much more rapidly through the membrane through protein channels called aquaporins. All right, and then the last part of 2.5, membrane permeability, here just talks about cell walls and how to provide a structural boundary. Okay, let's dive in here. Here's the same picture, but just knowing that it's selectively permeable. Anything that's charged will not be able to pass through these nonpolar tails here, the phospholipid, and anything that is small and nonpolar can float right through. So ask yourself this. For each of these molecules, how can it go through the membrane? Can it flow right through? That would mean if it's nonpolar and small, it can just flow right through. Or if it's polar or large, then it's going to have to use some type of protein channel like this. All right, so go ahead and give it a try. And hopefully you get nitrogen is nonpolar. It will be able to just diffuse right through. It shares its electrons evenly. Oxygen is nonpolar and can diffuse right through, which is good for us. That way we can get enough oxygen into our mitochondria. Carbon dioxide is nonpolar and it can flow right out. So that's how we get rid of waste. Large polar molecules such as sugars will need to use protein channels, even if it's in a higher concentration on the outside than on the inside. And you would think via diffusion would come in, it still must use a protein channel. Nonpolar big molecules can diffuse through, but they might take longer. These ions, such as a chloride ion, sodium ion, potassium ion, these are charged, and they must use their own channels. And they have channels specifically for them. Just like enzymes are specific, here they have channels that are specific for each of them. All right. Here we go. I'm going to quickly clear screen, and let's move on. Okay, the cell wall provides structural boundaries. It's just made up of complex carbohydrates. It's nothing that you need to memorize or anything, but it's unique in plant cells in that they can have water come in via diffusion and it won't explode. Right, the cell wall will push back with what we call turgor pressure. Animal cells don't have a cell wall, but it doesn't mean that there's just nothing on the outside. There is stuff on the outside. It's called extracellular matrix, and it's often collagen fibers. And these can hold together cells and connect them as well. All right, quiz yourself. This was a short one. Can you describe which items can cross the membrane freely and which cannot? And can you describe the role of the cell wall? The whole name of the game here, students, is selective permeability of what can come in and out. Okay, let's get into 2.6 and 2.7. I'm going to do them together. This is dealing with membrane transport. And so, I'm sorry, I had an animation glitch there. Give me 1 second. Thank you for your patience there. So this is going to be talking about passive and active transport, how items get into the cell either via diffusion or if they need to use energy to go against their concentration gradient. And then also large molecules, endocytosis and exocytosis. Notice how the objective here is that these are mechanisms that they use to maintain solute and water balance in their cells, right? So as a cell uses up materials on the inside, it would need to bring in more sugars to keep doing cellular respiration. It might need to move sodium and potassium ions in different places for action potentials, et cetera, to help maintain solute and water balance. All right, and then 2.7 or facilitated diffusion just dives a little bit more into it, dealing with diffusion using protein channels, and so aquaporins, charged channels, and then it's going to deal with active transport, which has to use a protein channel. We'll get into co-transport, which is secondary active transport, and the sodium potassium. All right, here is diffusion in a nutshell. So you come in for lunch and somebody sprays Axe or they spray perfume, right? And so it's going to go from an area of high concentration to low concentration. These particles are moving randomly, but because they're moving randomly, they tend to spread out throughout their container, right? Even though they're all clumped together here to start with, some of them might move in this direction, but overall, they'll tend to move randomly to then spread out in the container, which is what's kind of happening here in this permeable barrier. And that would be called diffusion. Eventually, diffusion reaches dynamic equilibrium, where the molecules continue to move around, but they're spaced out evenly. So when a cell is in dynamic equilibrium with water molecules still move in and out, but it maintains the same balance of water. Diffusion is affected by temperature. Items that are hotter, and so we do this in ninth grade biology, will diffuse faster. So when we put a drop of ink into hot water, here's a GIF of it, and so this one would be the hot one. It should be spreading quicker. All right, passive transport requires no energy, right? And it's because it's going from high to low concentration, like water flowing down a dam. Here, as we start this diffusion process, all these particles are here. These particles are nonpolar and are able to cross the lipid bilayer. Eventually, even though they're moving in random directions, they'll eventually spread out so that they're at dynamic equilibrium over time, right? And so here you can see it reached dynamic equilibrium via passive transport. Here, this is an example of passive transport using a protein channel. And so notice how there's more of these green dots out here. So that would be the high concentration, less in here, and it goes from high to low until it reaches dynamic equilibrium. In our cells, a good example of this would be glucose, right? And so a protein channel brings in glucose, but your cell is going to oxidize or use up that glucose. It's going to burn it up to make energy. So therefore, it should maintain a concentration gradient, or a high concentration outside and a low concentration on the inside. In many classes, you can do like an egg lab where you take an egg, you put it in vinegar, you dissolve the shell, and if you put it in distilled water, it is now permeable. Water will come into the egg. It'll make it bigger. It'll expand. And if you put it into corn syrup or some other syrup, it will shrink, right? Because that is the water will leave. It'll go to an area of higher solute concentration. And so that is what we're going to get into now. A solution that causes a cell to lose water is called hypertonic. A solution that is at an equal rate of water transfer or dynamic equilibrium will be called isotonic. And a solution that is more dilute on the outside and causes water to rush into the cell will be called hypotonic. All right, here's a good example. This would be a semi-permeable membrane that does not let the balls go through, but does let water go through. And you can buy these. They're pretty cool. They're U-shaped. And the water will actually go up on this side because water is going to move to an area of higher solute concentration. We'll go over that in just a minute. All right, let's look at this one. Take a second to read it. Okay, in this picture right here, one second while I get my pen. There's 75% water on the outside and 25% solute. Inside the cell, there's 45% water and 55% solute. The water goes from an area of high water concentration to low concentration. High water to low water. It will go into the cell. This is called a hypotonic solution. I remember that because it makes it bloat up like a hippo. Now, in reality, water moves. It does move from areas of high water concentration to low water concentration. But you could also think of it as moving from areas of low solute concentration to higher solute concentration, because it's going to go grab onto all those solutes. So here it's moving towards that greater solute concentration. Here, and we're going to learn about water potential later on. 90% water inside the cell, 10% solute, 63% water on the outside of the cell, 37% solute. The water is going to leave the cell. The cell is going to crenate, it's going to shrink, and this one, the cell expanded. Eventually, this cell could explode in a process called lysis. This cell will shrivel up, and this solution, we call it hypertonic relative to the cell. It has relatively less free water, and it has more solutes. So water is going to flow from high to low, or from areas of lower solute concentration to areas of higher solute concentration. So when the cell shrinks, hypertonic. When it bloats up, hypotonic relative to the cell. And the cells can't control this because the aquaporins are open. These protein channels are open. So our body makes sure to maintain our water balance by sending us hormones to drink or to go to the bathroom or whatnot. Here, and this is also important when you're making an IV for someone, this would be an isotonic solution. The water is going in and out at an equal rate because there's 50% water here, 50% water on the inside of the cell. It is at dynamic equilibrium and the same solute levels. All right, let's move on. We'll get more into this with water potential, and we'll talk about osmolarity, etc. Let's look at active transport. Sometimes we need to go against the concentration gradient. When we sprayed the perfume or somebody passes gas, it goes from an area of high concentration to low. It spreads out. Sometimes we need to go against that. We need to go from areas of low concentration to high concentration. For instance, here, if I look and I see the blue dots, the blue sodiums, we need to get as many of them out of the cell as possible. That's going to require energy in the form of ATP. So here's how the sodium potassium pump works. There is ATP, and it comes and it binds with the pump, and it pushes 3 sodiums out. The pump then can bring 2 potassiums in. Notice how both of these are going against their concentration gradients. You might wonder, why is this happening? And the reason is going to be is this is how action potentials work. So there's something called voltage-gated sodium channels. It's going to create an electric potential difference across the cell. This will be relatively more positive out here, and this will be more negative on the inside. We will deal with that later on as an illustrative example. So that's how the sodium potassium pump works. That requires lots of energy. It's about, I think it's between 30-50% of the energy that you use while you're asleep. It's just running this pump. All right. Sometimes there's secondary active transport where you can couple things together. For instance, we've already used energy to establish this sodium potassium pump, vitally important. Well, we have what's called a symport. And in this picture, there's a protein that allows some sodium back into the cell, but yet also brings a glucose molecule against its concentration gradient. So you could say that there might be glucose here in the in this extracellular fluid, on the fluid outside of the cell, right? Left over from when you ate something. There's relatively more glucose in the cell, but we want the glucose in here so that way we can burn it, we can use it to make energy, or maybe we'll store it in the form of starch as glycogen, etc. So this glucose symporter is an example of what we call co-transport. It's essentially using or hitching a ride off the sodium potassium pump. It still requires energy. This is still called active transport because the energy was used to establish the sodium gradient out here. It was used on the sodium potassium pump, the previous slide, and now it brings in a glucose molecule against its concentration gradient. This is a symport. They're going in the same direction, the sodium and the glucose. If they're going in the opposite direction, then it's called an antiport. So we can see that in the next one. Here is an antiport, sodium and calcium going in different directions. So the sodium's coming in with its gradient, right? The energy's already been spent to get the sodium out here. And the calcium is going against its gradient. This is important in cardiac muscle cells. So that is an example of an antiport. The symport would be the glucose and the sodium from the earlier one. I encourage you to draw this out, and I think that's a good way to learn it. Okay? All right. Endocytosis and exocytosis. This is infolding of the membrane in endocytosis to bring in food particles or to bring in something else. This requires energy, even though it's going from high to low concentration. Because of the infolding or the invagination of the membrane and that proteins will have to move it around, those proteins require energy. So this requires energy. It's active transport. Same thing with exocytosis, or this getting rid of something, getting it out of the cell, exiting it out of the cell from this vesicle. It requires energy to move it. So that's why we call it active transport. All right, why is this stuff important? Well, if you go to a grocery store and you see them spraying distilled water in vegetables, right? They're trying to create a hypotonic environment outside the plant cell. Plant cells will not burst because they have a cell wall. The water will go in and it'll make it look really nice and plump and rigid. Have you ever gargled salt water for a sore throat? That's creating a hypertonic environment around your throat cells to get rid of it if they're inflamed. And then there was a very sad case of a woman dying from a water drinking contest. And it's called hyponatremia. And so her blood became way too hypotonic relative to her cells, and it rushed into her brain. Because the skull is fixed, her brain expanded, and it actually led to her death. And then also the evolution of contractile vacuoles for these paramecium, right here for this amoeba, right? It's the inside here is going to have relatively more solutes than the outside, so water's going to come in. The water's hypotonic relative to the paramecium, and to prevent it from exploding, it needs to constantly pump water out. All right, quiz yourself. Can you describe diffusion, factors that affect it? Some factors we didn't go over were diffusion distance, but we'll do that in class. How materials are transported across cell membranes, active and passive transport, primary and secondary active transport. And that is it. And that is it for your summer assignment. Take care.